Hello guys, today I will be discussing about the myasthenia gravis pathophysiology, and I will try to make it more simpler. Myasthenia gravis is a autoimmune neuromuscular disease that it is a autoimmune neuromuscular disease that led to the increasing fatigue with sustained effort or repetitive effort. Take note of this one. Our own immune system produces the autoantibodies which are directed against the postsynaptic membrane protein in the neuromuscular junctions. These postsynaptic proteins are the acetylcholine receptors and the muscle specific tyrosine kinase, or the MuSK, which is present in the postsynaptic membrane, which is also known as the sarcolemma. In 85% of the patient with the myasthenia gravis, autoantibodies are produced against the acetylcholine receptor. In other 15%, remaining 15% of the patient with the myasthenia gravis, autoantibodies are produced against the muscle-specific tyrosine kinase. Autoantibodies against the acetylcholine receptors are normally immunoglobulin G type. It will cross-link the acetylcholine receptor. What do you mean by the cross-link the acetylcholine receptor? Actually, this is the this is the terminal knob of the neuron. Then this is the muscle sarcolemma. This actually called as the neuromuscular junction. After you will sensitize the neuron, or if you decided to command the muscles, then from the neuromuscular junction there is the acetylcholine. Then the neuron will release the acetylcholine, and to accept the acetylcholine, this is the sarcolemma that has this villi type of the appearance. This top portion is termed as the crest, and this has the acetylcholine receptors. Not only one, there are many acetylcholine receptors. Now, in the pathologic conditions, now your body produces the autoantibody. Now the autoantibody will go and bind this acetylcholine, make the cluster of the acetylcholine receptors. Now this cluster is formed. This is not happened in the normal persons. Since this cluster is formed, now what does the body will do, what does the muscle cell will do, is that the muscle cell will think this is the pathological condition, and muscle cell will pull this cluster inside the muscles. And now we all know that muscle cells has the lysosomes. Now this cluster is pulled inside. This has the autoantibody and the acetylcholine receptors. Now these both will fuse together, and this is the lysosome, and this both will form the phagolysosomes, and the muscle cell will destroy this one. The enzyme that is present inside the lysosomes is the hydrolytic enzyme that will destroy these vesicles, or these receptors, and the autoantibody all. This is the pathological conditions. It will cross-link the acetylcholine receptors, and pathological clustering of the receptors, as we saw. Cell will internalize the cluster, or the endocytosis occur, as we saw. Catabolism and destruction of the receptors with the help of the lysosomes. But in the absence of the autoantibody, endocytosis and the catabolism is the normal mechanism to replace the old acetylcholine receptor with the new acetylcholine receptor. Actually, if in the normal person also, it is the regular phenomenon to replace the old acetylcholine receptors with the new acetylcholine receptor. But with the patient, in the patient with the myasthenia gravis, there will be the formation of the cluster. That means in a single time, the many acetylcholine receptors is internalized and destroys. That is the pathological, and those cluster is made by the autoantibody. That's why those many acetylcholine receptor that has been internalized and destroyed should be replaced by the new acetylcholine receptors, and in pathological condition that could not happen, and the patient will have the less acetylcholine receptors in the neuromuscular junctions and manifest the disease. Okay, and those internalization of the old receptors and the replacing of the new receptor is the normal. Then what is the abnormal in the patient with the myasthenia gravis? In myasthenia gravis, autoantibody accelerate the endocytosis of the receptor and destruction of the receptors occur. Autoantibody will accelerate the endocytosis of the receptors and the destruction. That this autoantibody will accelerate, which does not occur in the normal people. Cell is unable to compensate and maintain the normal concentration of the acetylcholine receptor in the postsynaptic membrane. Now that will lead to postsynaptic membrane will become the severely deficient in the acetylcholine receptors, that will alter the or the decrease the neuromuscular transmission. Autoantibody does not stop there. Fc portion of the autoantibody, that is the immunoglobulin G, will become active. Fc portion means if this is autoantibody, this is known as the Fc portion of the autoantibody. This will become active. As we all know, this is bind with the acetylcholine receptor in the sarcolemma of the neuromuscular junction. In the neuromuscular junctions, this is the postsynaptic membrane, and this is also acetylcholine is bound there, and okay, Fc portion of the autoantibody IgG will become active. This will become active. Then after it will become active, that this will activate the complement system. That means MAC, that is the membrane attack complex, which is the part of the complement system. This will include C5b, C6, C7, C8, and C9. These all protein will make the structure like this one, like this, and this will be drilled inside the muscle membrane, muscle sarcolemma. This will be inserted inside, and this will make the hole. MAC will drill the cell membrane, and the membrane permeability is disturbed. Membrane loses the functions and the cell will die. Total surface area of the synaptic membrane is now reduced. This is the normal, and this is the patient with the myasthenia gravis. If you'll see here, then if you'll see the crest of the sarcolemma, this has a lot of the acetylcholine receptor. If you'll see here, in the pathology condition, they have the few. This red spot is the red structure is the acetylcholine receptor. If you'll see in the person with the myasthenia gravis, that it is very little few, and in normal condition they have the a lot. Total surface membrane loses the function and it will die. Total surface area of the postsynaptic membrane is now reduced. If you'll see the surface area, now it is reduced. It is a lot of surface area, now it is reduced due to the membrane attack complex. Due to the membrane attack complex making drill and destroying the surface. Density of the receptor per unit surface area is reduced, and increase in the synaptic gap. If you'll see, then you will see the increased synaptic gap, and here it's normal, it's not that much. Now this both the decrease in density, decrease in density, and the increase in synaptic gap, both will decrease the neuromuscular transmission, and the muscle will become weak. Another thing we need to take note, that not only the acetylcholine receptor, there are also the MuSK protein present in the sarcolemma or the postsynaptic membrane. This is the MuSK. Okay, what is the function of the MuSK? There we have discussed that the old acetylcholine receptor is normally replaced with the new acetylcholine receptors. No, this will be internalized and destroyed, and the new will be occupying the place of the old. Now this trafficking process is actually, this trafficking is controlled by the muscle specific tyrosine kinase, or the MuSK. Okay, in the 15% of the patient, there is the presence of the anti-MuSK antibodies. If there is the presence of the anti-MuSK antibody, it do not activate the complement system, but it will bind with the muscle specific tyrosine kinase. The antibody will bind with the muscle specific tyrosine kinase. Then what happens? Intracellular trafficking of the cholinergic receptor is disrupted, and concentration of the acetylcholine receptor in the postsynaptic membrane is decreased. Concentration of the acetylcholine receptor is decreased. If the new acetylcholine receptor could not replace the place of the old acetylcholine receptor that has been internalized and destroyed, then what happens is the concentration of the acetylcholine receptor in the postsynaptic membrane is now decreased, and that will result in the decreased neuromuscular transmission. Now, role of the thymic abnormalities, because we need to talk about the thymus, because there is the thymus role in the production of these autoantibodies. Only those patients who have the anticholinergic receptor autoantibody will have the thymic abnormality. 10% of the patient, because we have said that 85% of them will have the anticholinergic receptor autoantibody. And among the 85%, 10% of the patient will with the myasthenia gravis will have the thymoma. That is the tumor of thymic epithelium. Take note of the epithelial cell. This is the tumor of thymic epithelial cells. And among the 85%, 50 to 60% patient among 85% with the myasthenia gravis will have the thymic hyperplasia. That is B cell hyperplasia, and that will develop to the B cell follicles. Okay. Actually, the myasthenia gravis is bimodal disease. What do you mean by the bimodal? Bimodal means this will affect the two age group of people. That means in younger age, it will mostly affect the female. The female of usually the childbearing age, and that means in those female especially there will be the hyperplasia, not the thymoma. This will in the hyperplasia is usually common to see in the younger age and female. When we talk about the thymoma, this will actually occur in the old age, that is the male. Now let's talk about the relation of the thymic abnormality with the production of autoantibodies and having the myasthenia gravis. Normally the myoid cell in the stroma of the thymus do not express myoid cell. There is the myoid cell in the stroma of the thymus do not express the actually the myoid cell will express the cholinergic, not actually the cholinergic receptor. Cholinergic like receptor is expressed by the myoid cell to the B and T cell. Normally B and T cell do not recognize these proteins as foreign. If it will, then those cells will be suppressed or killed. Because when you will show the cholinergic receptor like protein to the B and T cell in the thymus, if the B and T cell will react, then most likely those B and T cell will react against all the neuromuscular junction acetylcholine receptor, and the thymus does not want that to be happen. So the thymus will suppress or kill those reactive B and T cells. But in the people with the thymoma or the thymic hyperplasia, thymus function is disturbed in such a way that when muscular antigen is presented to the B and T cell will react with them. That is the difference between the normal and abnormal. Now those B and T cell proliferate and produce the antibody. Actually, the B cell, specifically the plasma cell, that is the matured B lymphocyte plasma cell, will produce the antibody. Via the systemic circulation, the antibody goes to the autoantibody will go to the neuromuscular junctions, cross-react with the acetylcholine receptors and result in the myasthenia gravis. Cross-react that phenomenon that we have already discussed previously, and that will result in the myasthenia gravis. And normally this is the pathophysiology of the myasthenia gravis, and I hope you have learned a lot about the myasthenia gravis. Thank you.